guess following on from what Philo was talking this morning, he's going to tell us about circulating exosomes and the roles in ischemic heart to recruit bone marrow stem cells. Thank you, Gangjian.
So thank you, everyone. I'd like to start by thanking James and Jay for organizing this wonderful meeting, so also for the opportunity to listen to these wonderful talks and for the opportunity to actually also report our research. Okay. Okay, as we do all know that after ischemic injury that there are a massive amount of bone marrow cells that influx into the ischemic myocardium. And this large amount of bone marrow cells that would have a significant impact on the outcome of the disease process, as we are informed beautifully yesterday and today by Professor Dr. Harvey through this single cell analysis and lineage tracing studies that including a lot of bone marrow derived cells as well. It's just so slow. So we believe that bone marrow cells, that especially bone marrow-derived stem cells, progenitor cells, play a very important role, as shown by this very early paper a decade ago, showing that bone marrow CKD cells that when have a deficiency, the ischemic heart is doing very badly. And also that it looks like in human that circulating stem cells, CD34 cells, has very strongly correlated with the mortality of ischemic heart disease. And so one of my projects has been focused on this, how the ischemic heart mobilizes bone marrow cells, that how to inflammatory cells and progenitor cells into ischemic heart, how to exert the reparative effect. And through many years of study, we were able to actually establish that it looks like this become alpha-4 integrin and STF4 and 6L4 chemical and stem cell factor CKD receptor tyrosine kinase. These interactions cross-talk to each other, signals that seems to be the most important role actually to maintain the stem cells in the adult bone marrow. So you disrupt them, you actually mobilize stem cells. And also have been shown a few years ago that it looks like that this signaling, especially in that SXL4, seems to be very, very important for the mobilization of EPCs. And also we will focus a lot in the ischemic myocardium, how after these cells are migrated into the ischemic myocardium, how they signal migrate, invade actually in the ischemic tissue. Recently we move on actually study that how once the EPCs got into the ischemic myocardium, how their metabolic states actually affect their reparative capacity. For example, in this paper published last year, we were able to show that it looks like, as just shown by Dr. Fukuda, that it looks like oxidative metabolism in these EPCs seems to be very crucial for them to be able to adapt the endothelial phenotype to be differentiating into endothelial cells. And so today I'm going to focus a little bit more on a new finding we have just made recently in the lab, that we looked carefully how actually ischemic injury could signal and mobilize bone marrow progenitor cells. And this has been performed by this group of people in the lab that Dr. Chin is a former trainee and postdoc and the PhD student in the lab. So we are particularly focused on microRNAs and exosomes. The reason that we're doing that because we know that this particular cardiac and skeletal muscle specific microRNAs expressed a very high abundance in the skeletal muscle and in the myocardium, and MYO208, MYO499-5P, and MYO1 and 133A. And also, once myocardial infarction occurs, a large amount of this microRNA got into the circulation and dramatically downregulated in the heart themselves. And as you can tell from these clinical studies, in my patients repeatedly that this seems to be the major microRNA released into circulation from the ischemic heart. So they are considered as biomarkers of this disease. And also exosome, as we all know that, also from Dr. Monach's work that showing the stem cells that create a lot of exosomes have a lot of beneficial effect, and also vice versa, that this microvesicles that generated through the endosomal pathway get released into circulation and actually mediate the exchange of substances like proteins, RNAs, DNAs, and lipids that mediate this cell-cell communications and also between different organs. And so this seems quite to be interesting. So we actually, firstly, we made a myocardial function in the mice. Then we analyzed this particular myocardial microRNA. We find that these microRNAs are exponentially upregulated in the circulation in the blood. And we want to be very careful, so we performed the microRNA sequencing study. We were able to actually compare the normal heart and the myocardial infarction heart, and actually be able to identify these 12 microRNA is actually a very relatively stable, irregardless of the myocardial infarction or not. So these are very good microRNAs for us that stably expressed to normalize our analysis, which give us some very precise results. And we find that also after myocardial infarction, we at 12 hours, we analyzed this microRNA in different organs, and they are supposed to be released only from the myocardium. We find that the majority of the increase, a lot in the bone marrow is different microRNAs in the bone marrow mononuclear cells or not. And some a little bit in the kidney, but not much difference in the spleen and in the liver. And also the change of differences is very big. So this seems to have a certain selectivity to the bone marrow. We don't know yet whether how they get in there more preferentially. And also that this is not because of the upregulation of this microRNA in the bone marrow during the myocardial infarction, because that if we culture these bone marrow cells in the hypoxia and normoxia, there is no change compared to the positive control of cardiomyocyte. And also measure the dynamics of bone marrow cardiac microRNA in a time course study. We find that after myocardial infarction, it goes very high in the blood, then goes quickly down, pretty much return to the baseline at 3 days. So then we study whether they are carried in the exosome. So basically we isolate a very tiny mouse blood in the plasma, and very carefully analyze and characterize these exosomes. And we find that we analyze the component in the exosome and the component that in the plasma, but depleted of exosome. We find that the seems this 1A, 2OA, and 399 are predominantly exist in the exosome, and also this 133 seems to have a higher level in the non-exosome component. And then we actually when studied whether this exosome is the reason that mediates this microRNA transfer into bone marrow cells. So we can perform the co-culture assay of bone marrow mononuclear cells, apply to the exosomes, and we isolate from the plasma of mice with or without myocardial function. We find that in this culture study, we find that irregardless of the component, we only have the most of it. Only that exosome component actually increased the co-culture, the BM mononuclear cells have increased microRNA, but if non-exosome components doesn't really change the level of the BM of the exosome in the bone marrow mononuclear cells, suggesting that it looks like this exosome transfer the microRNA more efficiently than the non-component microRNAs, non-exosome microRNAs. And also that we inject the exosomes from isolated from the plasma of myocardial function mice, and we find that also after about 12 hours, we then harvest the recipient which do not have MI. We find actually the microRNA level is increased, so it's actually kind of physiologically happen. And in fact, that the CM exosome and the myocardial function exosome, when we label with a chemical dye injected into circulation, we then analyze the bone marrow mononuclear cells. This seems to get themselves into the bone marrow cells equally efficient. And also, we collaborate in collaboration with our cardiology group. We find that we compare the exosome containing microRNAs in the patient that with the acute ST elevation myocardial function versus those patients actually without any sign of acute coronary syndrome. We find that in this myocardial function basically making the circulating exosomes contain way much more microRNA. So still again suggests that this microRNA actually are contained in the exosome of patient. So what's the molecular target? We have performed a lot of bioinformatics analysis, and basically one of the striking features is that we by screen different software platform, we find that basically that we consistently found that CXCR3 UTR contains MIR1 actually binding site. So we were able to confirm that MIR1 basically that regulates, down-regulate expression of this CXCR4 in a reported assay, but the other three microRNA didn't. So, but however, when we overexpress these four microRNA in the bone marrow mononuclear cells in vitro, we find actually exclusively down-regulate CXCR4. So it looks like the other ones down-regulate CXCR4 through different mechanisms. We don't know why. We actually also repeated this in the bone marrow-derived mesenchymal stem cells. Also, they do the same thing. And then this from the protein analysis, FACS analysis, consistent. And also not all the microRNA. Actually, we find this very closely related skeletal muscle microRNA. These two particularly didn't regulate the CXCR4. So there's some specificity there. And then whether this is mediated by exosome. So basically that we again isolate the acute myocardial infarction exosome versus a sham exosome. Actually, we culture them in the bone marrow mononuclear cell culture. They were able again to down-regulate the CXCR4. But if we use antagonist pre-treatment of mice before we isolate the exosome, and this actual down-regulation is actually mitigated. So still again suggests this is mediated by this microRNA. And also we perform this in vivo. Again, that we injected the mice, the intact mice, with this exosome isolated from myocardial function. We were able to see that there is a CXCR4 down-regulation in the bone marrow mononuclear cells after 12 hours. And actually, if we pre-treated the donor mice with the atagomia, then this exosome no longer be able to downregulate self-expression in the recipient bone marrow mononuclear cells. And we actually we control our experiment very well because of that there's other two very well-established target of this microRNA actually are able to downregulate it, and then this antagonistic treatment are able to restore the expression. So basically, we believe that our antagonistic treatment of the donor and also the experiment actually controlled okay. So then lastly, we look at whether there is any biological significance. So basically, we look at the progenitor cells mobilization. And particularly that we perform the FACS analysis. We find that these lineage-active cells and these lineage-secret positive cells, and also these secret positive lineage-active cells in the blood circulation is actually increased when the intact mouse are treated with exosome from myocardial infarction mice. Actually, if the donor pre-treated with the antagonism, this effect is no longer there. So this suggests that this really, and also we perform this hematopoietic stem cells colony assay in the recipient blood. We find that actually it's the same conclusion that if the mice treated with myocardial infarction exosome, they have a mobilization of colony forming cells. Actually, this is blocked by if you pre-treated the mice, the donor mice with the antagonism. So basically today I reported to you that after myocardial infarction, this myocardial-specific microRNA, including these four, are actually elevated in the circulating exosomes, and these are carried primarily in the exosomes and also mediated and the bone marrow mononuclear cells, CXCR4 expression, and actually these have seems to be able to mobilize bone marrow stem cells into the circulation, and that's so basically. I also cited this paper published in, quite a few years ago by Nosodo, showing that it's a kind of a review paper showing that where we demonstrate the bone marrow progenitors, we have some microRNA, or maybe on the other hand, actually the cardiac may also talk back to the bone marrow actually in this form, maybe a kind of a full cycle of life. So with that, I'm going to end my talk and thank people in my lab, and I already acknowledged the people who does the job, and thank NIH funding, and thank you for your time.
We'll take one quick question, Bob.
Very nice studies. So after an infarct, you also turn on your sympathetic nervous system markedly, and that also alters the efflux of cells from the bone marrow, and tethering of stem cells in the marrow to the niche are controlled by adrenergic mechanisms. Have you looked to see what happens if you block the adrenergic nervous system after infarct, which we commonly do in patients, to see whether that alters the ability of stem cells to come out into the circulation?
So you're talking that this CXCR4 inhibition also affect the influx of cells?
Adrenergic mechanisms regulate the efflux of stem cells from bone marrow very markedly. So I was wondering if you'd get rid of that and see whether you still see the same degree of efflux, because we commonly use beta blockers, for example, after infarction.
Okay. So okay. Yeah, that's a good question. Yes, beta signaling, especially through the beta 3, that actually play a very important role, and our model is a very sympathetic model. But however, we isolated the exosome from the infarct mice and purified exosome and inject back. So this perhaps would actually exclude the possibility.
I was trying to understand the relative contributions.
Oh yeah. That's so we believe that there are so many. The significance of this work is that now we kind of try to identify there is a non-soluble, non-nervous mediated mobilization process. We have no idea what's the relative importance of this versus others. Maybe less relevant because recently, actually, we are working on that. What we are doing now is that we develop a way of specifically enable the cardiac exosome. And only look at, and without giving exogenous exosome, just look at how endogenously this does the work. So this would give me a little bit more resolution for the relative importance of different pathways.
Okay. Thank you, Gangjian. So